In this video, we're going to cover the structure of the synapse and the mechanisms neurons use to send signals across it. We're also going to take a look at the different types of neurotransmitters and receptor proteins. If you haven't seen the previous lecture on action potentials, I highly suggest watching that first. Now let's start this lecture by first defining what a synapse is. A synapse is a specialized junction or a meeting point between two neurons or between a neuron and a target cell like a muscle or a gland, where information is passed from one neuron to the next. In order to stimulate or inhibit the activity of another cell, one neuron communicates with it at the synapse. It serves as the channel for communication, as this is how neurons interact with one another or talk to one another. There are more than 100 trillion synapses in the central nervous system. If you've seen the lectures on neuron anatomy and action potentials, you know that synapses form between axons and dendrites and consist of a presynaptic neuron, a synaptic cleft, and a postsynaptic neuron. The synaptic cleft here is the extracellular space between these two cells. And the electrical activity of a presynaptic neuron influences the electrical activity of a postsynaptic neuron. So when an action potential sends an electrical signal to the end of an axon, the signal travels to a synapse where it's converted into a different type of signal and sent to another neuron. The transfer of electrical signals at synapses influences whether the postsynaptic neuron will fire its own action potential. Okay, now there are two types of synapses. We have electrical and chemical. Synaptic transmission can either be electrical or chemical. So now let's subtract complexity and break down the differences between these two, starting with electrical synapses. At electrical synapses, there is a direct physical connection between the presynaptic and postsynaptic cells, and this connection is called the gap junction. Let's zoom in. Gap junctions connect the plasma membrane of the pre- and postsynaptic cells. These synapses contain protein channels that allow charges and ions to flow from one cell to another with little or no delay. Because of this, electrical synapses have the advantage of enabling very quick cell-to-cell communication and allowing for the synchronized activity of groups of cells. Examples include both cardiac muscle and smooth muscle, because when it comes to a beating heart, we need an extremely rapid and synchronized conduction so that the contraction of the heart is coordinated. Okay, so electrical synapses transmit signals more rapidly than chemical synapses do. Now, if there's an advantage, what are the disadvantages? Well, this type can't convert an excitatory signal in one neuron into an inhibitory signal in another. They don't have the adaptability and capability to modify signals that we see in chemical synapses, okay? And this leads us to the structure of the chemical synapse. When it comes to chemical transmission, this is where chemical messengers known as neurotransmitters come in. What they do is carry information from the presynaptic neuron to the postsynaptic neuron. Let's break this down. Recall that synapses are formed between nerve terminals and the dendrites or cell bodies of the other neuron. A single neuron can receive hundreds, thousands of synaptic inputs from many different presynaptic neurons. So right here, we have the axon of the presynaptic neuron, and it'll come down to form this terminal button. So this is the presynaptic neuron, and this right here is the presynaptic membrane. And the other one here is the postsynaptic neuron, and this is the postsynaptic membrane. The synaptic cleft, again, is a small gap between the pre- and postsynaptic neurons that is about 10 to 20 nanometers in size. This is what separates the presynaptic and postsynaptic neurons. And a neurotransmitter that is released from the presynaptic axon terminal is used to transport signals across the synaptic cleft, and this is what will bind to the postsynaptic receptors. Occasionally, more than one neurotransmitter is released, and these are called co-transmitters. And these neurotransmitters have different receptors on the postsynaptic cell, and this is one of the key benefits of chemical synapses, which allow for the integration of many signals that arrive at a single cell. Absolutely beautiful. Let's take a closer look at what happens here. There are small sac-like structures called synaptic vesicles, which contain neurotransmitter molecules, these green circles here. Before these neurotransmitters are activated, many vesicles dock at release regions known as active zones on the presynaptic membrane, while others are scattered. The question we now have is, how do we release neurotransmitters into the synaptic cleft? There are two factors that are important for the release of these neurotransmitters from the axon terminal. Number one is the depolarization of the terminal, the electrical impulse that travels down the axon. And number two is the presence of calcium ions in the extracellular fluid, okay? What I'm going to do now is I'm going to clear up some of these labels and let's go through neurotransmitter release step by step. So the membrane of the presynaptic terminal contains voltage-gated calcium channels. Recall that there are sodium and potassium channels in the neuron, but there are also voltage-gated calcium channels. And when an action potential reaches the axon terminal, this is going to stimulate the opening of voltage-gated channels, voltage-gated calcium channels, allowing calcium to diffuse into the terminal because the concentration of calcium is much higher outside the neuron than it is inside. So the depolarization during the action potential opens these calcium channels, okay? Once the calcium is in, what happens then? Well, the vesicles are mobilized by calcium to get ready to release neurotransmitters. Let's zoom in to how calcium triggers this process. All right, there's a protein called synapsin, which connects vesicles to the cytoskeleton of the cell. And calcium activates an enzyme that causes synapsin to dissociate from the vesicles, mobilizing them for release. Now recall that we previously mentioned that vesicles dock in the active zones. And these vesicles are docked or attached by the interaction of a number of proteins known as snare proteins. So some of these proteins are attached to the vesicle membrane and others are present in the membrane of the terminal. Synaptobrevin is a snare protein found in the membranes of vesicles, while syntaxin and SNAP25 are two snare proteins found in the cell membrane. These proteins form a complex that helps bring vesicles into contact with the cell membrane, allowing the two membranes to fuse together. Beautiful. Now, when calcium levels in the cell are high, these calcium ions bind to another type of protein found in synaptic vesicles. There's another type, and these are called synaptotagments. And we can view synaptotagments as a calcium receptor. So what it does is it's going to activate a conformational change in the snare complex that leads to membrane fusion and neurotransmitter release. It allows these synaptic vesicles to fuse with the axon terminal membrane, releasing transmitters into the synaptic cleft. And this occurs via exocytosis. After we've released neurotransmitters, the snare complex is disassembled and the vesicles is recycled so it can be used again. How cool. Now going back to these neurotransmitters, once they are released from a presynaptic axon terminal, they diffuse across the cleft and influence the activity of the postsynaptic cell. Okay, but before we break down how to do that, let's do a quick recap of everything we've just covered. So first up, an action potential reaches the axon terminal and depolarizes the membrane, which then activates voltage-gated calcium channels in the cell membrane. Calcium ions then flow into the terminal, which stimulates synaptic vesicles to do what? Release neurotransmitters. These neurotransmitters diffuse across the cleft and bind to receptors on the postsynaptic cell. And this leads us to how neurotransmitters influence postsynaptic cell activity. Okay, let's go through this. Neurotransmitters act by binding to receptors on the membrane of the postsynaptic cell. Their binding to the receptor is short and reversible. Some neurotransmitters are considered excitatory, that is, they increase the likelihood of a target neuron firing its action potential. Other neurons are viewed as inhibitory, having the opposite effects, making a target neuron less likely to fire an action potential. Sometimes a neurotransmitter can have either an excitatory or inhibitory effect. We're going to expand on this in just a minute. Now, typically, a single neurotransmitter can bind to and activate various different receptor proteins. The presence of a certain neurotransmitter's receptor on the postsynaptic cell determines whether its effect is excitatory or inhibitory at specific synapses. Now, we can divide the receptors that are activated by neurotransmitters into two main groups. We have ligand-gated ion channels, also known as ionotropic receptors, and then we have metabotropic receptors. Ligand-gated ion channels are large protein complexes. These are ion channels that open directly in response to ligand binding. Once a neurotransmitter binds, okay, we have the neurotransmitter here. Once these neurotransmitters binds, these complexes change shape, causing the channel to open and letting ions from the extracellular fluid enter the postsynaptic neuron. And this change in ion concentration can generate another action potential that can travel down this postsynaptic neuron. Okay, that's what ligand-gated ion channels are. The other group are metabotropic receptors, and these receptors are not ion channels. Instead, these receptors indirectly affect ion channel opening and closing. So when a neurotransmitter binds to these receptors, it will trigger a signaling pathway that may indirectly open or close channels. So these receptors influence ion channels through a G protein or a second messenger pathway. There are more steps involved with metabotropic receptors because several molecules have to be activated. But looking at this, in either scenario, when a neurotransmitter binds to a receptor, specific ligand-gated ion channels in the postsynaptic membrane open or close, which then leads to changes in the membrane potential in that neuron. Okay, and this change can either make the target cell more likely to fire its own action potential or less likely to fire an action potential. Let's break this down further. Okay. There are two types of chemical synapses. We have excitatory and inhibitory. The postsynaptic reaction to the neurotransmitter at an excitatory chemical synapse is a depolarization, okay? Making the postsynaptic cell more likely to fire its own action potential. We're bringing the membrane potential closer to the threshold for firing an action potential. Let's go through an example using glutamate, which is an excitatory neurotransmitter. So we have this beautiful blue glutamate here, and this glutamate will bind to this receptor, causing ligand-gated ion channels to open. Sodium will flow into the cell, making the inside of the membrane less negative than at resting potential. What this means is that it's going to bring this postsynaptic neuron closer to its firing threshold. And this change in membrane potential is called an excitatory postsynaptic potential, or EPSP. EPSP raises the postsynaptic neuron's membrane potential towards the threshold, making the inside of the cell more positive. Here's the thing. Sometimes one EPSP isn't enough to reach the threshold in the postsynaptic neuron, but when combined with other EPSPs, it can cause an action potential. We're going to break this down further in just a minute. But that's excitatory chemical synapses. Let's look at inhibitory synapses. At inhibitory synapses, the potential change has the opposite effect. It's hyperpolarizing, meaning it makes the postsynaptic cell less likely to fire an action potential. This is called an inhibitory postsynaptic potential, IPSP. IPSP decreases the likelihood of firing an action potential because the membrane potential is below thresholds. Okay, EPSP increases the likelihood of the neuron firing an action potential. IPSP, inhibitory, decreases the likelihood. Okay, and IPSPs can also neutralize or cancel out the excitatory effects of EPSPs. An example of an inhibitory neurotransmitter is GABA, and the binding is going to cause potassium ions to leave the cell and chloride ions to enter the cell, making the inside of the cell more negative. Recall from the previous lecture, we covered how when potassium leaves the cell, potassium ions, it's going to leave behind anions inside the cell, making the inside of the cell, more negative. All right? So the postsynaptic cell integrates all of the excitatory and inhibitory inputs, and an action potential is triggered when there are enough excitatory synaptic events to reach threshold. Now, we did cover spatial and temporal summation in the previous lecture, but for a refresher, spatial summation is the integration of postsynaptic potentials that take place at various sites, but roughly at the same time. Whereas temporal summation is the integration of postsynaptic potentials that take place in the same location, but at slightly different times. Okay, that's how summation works. So we've covered how neurotransmitters are released, how they bind to receptors on the postsynaptic cell, and how they influence the activity of the postsynaptic cell. What's next? Where do we go from here? Now we need to terminate the signal and allow the postsynaptic to recover to its normal resting potential and get ready for new signals. And this happens when neurotransmitters are released from their binding, and unbound neurotransmitters must also be removed to terminate the signal. Why? Why do we want to do this? Because we don't want the neurotransmitter to diffuse out of the synapse where nearby cells might be affected. Okay, there are a few different ways to do this, to terminate the signal, to remove the neurotransmitters. Number one, they diffuse away from the receptor sites. Okay. 2, they are actively transported back into the presynaptic axon terminal via reuptake. Another way is that they are transported into nearby glial cells where they are degraded or enzymatically converted into inactive substances, some of which are transported back into the presynaptic axon terminal for reuse. Now, the enzymes involved in this process may be found in the synaptic cleft, on the presynaptic or postsynaptic membrane, or both. All right. So we've outlined the process of how neurotransmitters are released, how they interact with the postsynaptic membrane, and finally, all the different ways they are removed from the cleft, from the synaptic cleft. Let's now move on to the factors that determine synaptic strength, because both presynaptic and postsynaptic processes affect how strong or effective a synapse is. Let's go through presynaptic factors first. The first one, this is quite straightforward, neurotransmitter availability, including the availability of precursor molecules as well as the activity of the rate-limiting enzymes in the neurotransmitter synthesis pathway. Another reason is the concentration of calcium ions, if the calcium concentration in the terminal is higher than usual. This could be due to calcium removal not keeping up with calcium entry. So the amount of neurotransmitter released with subsequent stimulation will also be higher than usual, which can happen during high-frequency stimulation. So then the number of ion channels that open in the postsynaptic membrane and the amplitude of the EPSP or IPSP in the postsynaptic cell increases in proportion to the amount of neurotransmitter released. That is, the more neurotransmitter there is released, the more ion channels that open in the postsynaptic membrane. Okay? So these are just some of the presynaptic factors that determine synaptic strength. What about postsynaptic factors? One factor is the postsynaptic membrane's previous electrical state, either excitatory or inhibitory summation. Secondly, different receptor types use different signal transduction mechanisms, which can have varying effects on the postsynaptic cell. Also, the ability of a specific receptor to react to and respond to its neurotransmitter can change. This can lead to what's known as receptor desensitization, where a receptor initially responds normally when exposed to a neurotransmitter, but eventually stops responding despite the neurotransmitter still being present. This is part of the reason why drug abusers sometimes develop a tolerance to drugs that increase specific neurotransmitters, forcing them to take higher dosages of the drug to get the desired effects. Okay? Another factor that can alter synaptic strength is drugs and diseases. So a drug may increase the outflow of neurotransmitters from vesicles to the cytoplasm, exposing them to enzyme degradation, enzyme breakdown. It also has the potential to either increase or decrease neurotransmitter release into the synaptic cleft. It may also bind to a receptor on the postsynaptic membrane to block or mimic neurotransmitter activity, or even trigger or inhibit second messenger activity on the postsynaptic cell. But these are just a few presynaptic and postsynaptic factors that determine synaptic strength. Okay. There's one more thing we need to cover, and that is the different types of neurotransmitters. So far, we've outlined how neurotransmitters are stored in synaptic vesicles and how they get released when calcium enters the axon terminal in response to an action potential, and they act by binding to receptors on the postsynaptic membrane. Let's now talk about the two chemical messengers, neurotransmitters and neuromodulators. The main difference is that neuromodulators often alter the postsynaptic cell's reaction to particular neurotransmitters, enhancing or reducing the strength of ongoing synaptic activity. They change how well the synapse functions, so they might change the synthesis, release, or reuptake of a neurotransmitter. And when it comes to receptors for these messengers, neurotransmitter receptors influence ion channels, whereas receptors for neuromodulators function via G proteins coupled to second messenger system. Okay? Now, there are many substances that have been identified as neurotransmitters or neuromodulators, so we're only going to go through some of them, starting with acetylcholine. Acetylcholine is a key neurotransmitter in the peripheral nervous system at the neuromuscular junction, where a motor neuron connects with a skeletal muscle cell. Neurons that release acetylcholine are called cholinergic neurons. And there are two main groups of cholinergic receptors. We have nicotinic and muscarinic. The nicotinic acetylcholine receptor contains an ion channel, a ligand-gated ion channel. This receptor is a channel protein that opens to allow the passage of cations, both sodium and potassium. And nicotinic receptors in the brain play an important role in cognitive function and behavior, as well as learning and memory. On the other hand, the muscarinic receptors are metabotropic and coupled with G proteins. So recall that this is a membrane protein that, when activated by a neurotransmitter, indirectly results in the opening of ion channels by means of a second messenger. So these change the activity of several enzymes and ion channels. Okay? The next group are amino acid neurotransmitters. So first up, we have glutamate, which is a very common excitatory amino acid neurotransmitter in the central nervous system. Another one is glycine, which is a key neurotransmitter released from inhibitory interneurons in the spinal cord and brainstem. These bad boys bind to ionotropic receptors, allowing chloride ions to enter and decreasing the likelihood of an action potential firing. And then we have GABA, which is the major inhibitory neurotransmitter in the brain. Now, GABA is not one of the 20 amino acids that's used to build proteins, but it's still grouped with the amino acid neurotransmitters because it is a modified form of glutamate. The next class are biogenic amines, which are small charged molecules with an amino group that are synthesized from amino acids. Common ones are dopamine, norepinephrine, serotonin, and histamine. Dopamine, norepinephrine, and epinephrine are called catecholamines because they all contain a catechol ring, okay? This 6-carbon ring with 2 adjacent hydroxyl groups, as well as an amine group. Dopamine is a precursor of norepinephrine, which is a key neurotransmitter in the nervous system. And epinephrine and norepinephrine are synthesized in the adrenal glands and secreted into the bloodstream. They act to increase the heart rate, blood pressure, and levels of glucose. We also have serotonin, which is produced from an essential amino acid, tryptophan. Okay, it functions as a neuromodulator because of the gradual onset of its effects. Serotonin generally has an excitatory influence on the pathways that control muscles. So what it does is it prepares muscle cells for an excitatory response to other neurotransmitters. Next up are neuropeptides, which consist of 2 or more amino acids linked together by peptide bonds. Neurons that release 1 or more of the peptide neurotransmitters are called peptidergic, okay? And neuropeptides typically function as neuromodulators when they are co-secreted with another type of neurotransmitter. But compared to catecholamines, the amount of neuropeptide released from synaptic vesicles is considerably less. And it can interact with either ionotropic or metabotropic receptors after being released. Examples include endogenous opioids, which are a group of neuropeptides that includes beta-endorphin, dynorphins, and oncophalines, and their receptors are the site of action of opiate drugs like morphine and codeine. These are strong analgesics, that is, they reduce pain without making a person unconscious, and these drugs play a role in regulating pain. The next 3 groups are gases, purines, and lipids. Starting with gases, some short-lived gases also function as neurotransmitters. But here's the thing, gases are not released by presynaptic vesicle via exocytosis, nor do they bind to postsynaptic plasma membrane receptors. Instead, enzymes in axon terminals produce them in response to calcium entry, okay, which then diffuse into the intracellular of other effector cells where they bind and activate proteins. For example, nitric oxide activates guanylyl cyclase, okay, and this enzyme raises the levels of the second messenger cyclic GMP, which can change the postsynaptic cell's ion channel activity. All right. Next up are purines, ATP and adenosine, and these act as neuromodulators. And finally, lipids, which commonly act as neuromodulators. For example, we have prostaglandins. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video, or you can view the entire playlist. Hey, stop procrastinating.